Hi everyone. In today's class, we are going to discuss the most important topic in Unit 3, that is electromyography. In last class, we discussed what is electroencephalography and electrocardiography. Similar to that, in this class, we are going to deal one of the basic and as well as the major equipment, that is electromyography. So I think you might know what is mean by electromyography. So the name stands electromyography in the sense myo in the sense muscle, electro means electrical. Okay, so it means that this is the particular technique where we are going to record and also evaluate the electrical activity of the muscles. Okay, so as like a ECG and EEG, how we measured their electrical activity of brain and heart, here we are going to record the electrical activity of our muscle. That is the main purpose of having electromyography. So the term the electromyography and electromyogram, I think you might know because already we have discussed in previous class, right? So electromyography is a technique and graph is an instrument and gram is the output that you get when doing this experiment. Okay, so here you know one concept that when our cell is in resting state, it doesn't create any potential, right? So the cell have a combination of all the ions. So the cell membrane potential, when it get varies, the action and the ion movements, that is the intercellular ions and the extracellular ions concentration will get vary. So that variation will create some action potential. Okay, that is the basic concept. So once again I'm repeating, the cell usually is slightly negative in nature at resting state. When it get disturbed, the cell ions movement will take place. Because of that, the cell ions inside will get more, that is a positive ions compared to the negative ions outside. So the once it gets stimulated, the cell inside will becomes positive in nature. So that potential variant, that difference of ions concentration from inside and outside will generate one difference, that is nothing but action potential. Okay, the same concept here also we are going to see, because the group of cell is nothing but muscle. Here we are going to record the electrical activity of the muscle. So initially the muscle at its resting state doesn't create any action potential. So when you are measuring AMG, in that case means you can't see any potential variation. So for that, the requirement of doing this particular experiment is that we have to give some stimulation, sort of some stimulation to the muscle. Then only we will have a generation of electric potential. That electric potential we easily capture by means of this EMG machine, and we will analyze what is the status of that particular muscles. So this how this electromyography will functions. So now we will see what is the equipment aspect we need, or it is recommended when we are measuring the EMG. So first and foremost is electrodes, because once the stimulus being given, whether the stimulus we are giving from outside, that is externally, or by voluntary contraction or voluntary action, the muscle will get excited. So now it's being produced action potential. Now the thing is, we have to capture that action potential, then only we do further analysis process. So for capturing that action potential, then one particular component it's very much needed is electrodes. So that is the first and foremost, the electrode, that there are various types of electrodes. Here we are concentrating morely on two types. One is surface electrode, another one is needle electrodes. Okay, in the either of these two, we can use any type of electrode based on the demand or based on the application, whatever it is needed. Okay, so surface electrode means, you know that we are just placing over the surface, and after applying the gel, we will place the surface electrode and we will capture the action potential of particular targeted area. Or needle electrode means, then we have to make direct contact with muscle by means of needle. So that type of electrode is electrodes. Okay, so there are these two options we have for choosing the electrodes. One is surface electrodes, another was needle electrodes. The surface electrode is gives very generalized information. So if you need even more detailed clarity about the particular electrical activity of muscle, then you have to go with needle electrodes. So again based on the demand or based on the application, we can use any type of these two electrodes. Once the electrode has been fixed to capture the action potential of electrical activity, next we have to go with amplifier. The main purpose of the amplifier is that the whatever electrical signal we are capturing from our body biological system, it is always in a low in amplitude. That is, it's almost weak signal. So that signal is not easy enough to process by other electronic systems. So for that we have to make that particular electrical signal to some extent to great like what to strengthen that signal to some extent, so that it will be processed further in next stages. For that only we are using different types of amplifiers. We have low pass filter, like instrumentation amplifier, we have differential amplifier, or we have power amplifiers. There are n number of amplifiers there. So based on the like requirements or the type of amplification we need, we have to go with that particular amplifier, so that the input targeted signal raw signal will get amplified, it will become strengthened to go to process into the next level. Okay, now third and foremost is the output part, that is the connected to an oscilloscope. So first part is gate over, that is electrode, you capture the signal. Second, that captured raw signal you have to be strengthened by means of amplifying, that is the second part. And third part is connected to the oscilloscope, that is the output part. Okay, so this oscilloscope is it will be showing the electrical activity of your targeted part. So it will be like some waveform. From that waveform you can have some different type of analysis to identify the status of that particular muscle part. Okay, and not only that, you have other options of having audio unit, where the EMG signal will be fed to the audio unit, so that one can feel the spot, okay, spot of the signals. That is a one option. And next option is like even have a visual mode, that is one is oscilloscope, second one is audio mode. And you have some other design consideration you have to do, in order to remove some kind of interference. For that you have to go with some isolation circuit. Okay, these are the basic things you have to design or you have to consider when you are working on equipment. First is electrode, second one is choosing that type of amplifiers, third one is the what type of display unit you are using, that is oscilloscope or CRT, whatever it is. And fourth one is you have additional features of visual hearing, the hearing by means of audio unit, so that one can feel the part of that signals. And last one, for everything to be fixed in that proper manner, you have to be like do some isolation, so that you can avoid some kind of interference, that is electromagnetic interference. Okay, so this is all about the equipment part. So in next class, in subsequent class, we will be discussing even more about electromyography. Thank you.